Buonasera e benvenuti e benvenute. Iniziamo questa splendida serata che fa parte delle iniziative che abbiamo portato avanti con il progetto Vitality. Vitality è uno dei progetti del PNRR, uno dei progetti coordinati dall'Università degli Studi di Perugia. L'oggetto del progetto, il tema, è lo studio dei materiali avanzati, con particolare riferimento a due classi di materiali, i cosiddetti biomateriali, cioè i materiali che usano origine biologica, e poi i nanomateriali. Ecco, oggi siamo, per parlare di questa seconda categoria. I nanomateriali sono materiali molto particolari e oggi all'interno di questa categoria parleremo delle nanomacchine. Lo vedremo tra un minuto. Prima di presentare l'ospite molto prestigioso che abbiamo questa sera, vorrei chiedere al professor Perugini, che è il direttore del Dipartimento di Fisica e Geologia, che è il dipartimento che coordina il progetto Vitality, di dire due parole a nome dell'Università.
Si sente? Grazie. Per me è certamente un onore e un privilegio oggi portare i saluti del Dipartimento di Fisica e Geologia e accogliere il professor Jean-Pierre Sauvage, premio Nobel per la chimica 2016. Il premio Nobel è un riconoscimento straordinario che però prevede un'idea che porta a un risultato così eccezionale. E come nasce un'idea? Un'idea va coltivata in un luogo, va coltivata in una comunità. Ecco, vi potrei raccontare tante cose del Dipartimento di Fisica e Geologia, che siamo un dipartimento d'eccellenza, che abbiamo un budget plurimilionario, che stiamo crescendo, ma vorrei parlare più con il cuore che con la politica, e cioè invitarvi in dipartimento e farvi vedere quello che succede tutti i giorni. Voi potrete incontrare persone per i corridoi che parlano da sole, qualcun altro che prende a martellate una roccia, altri che girano con un visore tridimensionale esplorando un pianeta extraterrestre. Ecco, non poniamo limiti alle idee che possono essere sviluppate, coltiviamo un'anarchia culturale che è il sale della ricerca, e credo che il professor Sauvage potrebbe essere d'accordo nel dire che le idee non hanno bisogno di confini, non hanno bisogno di vincoli e vanno coltivate liberamente, in qualsiasi posto, che può essere un dipartimento, che può essere un viale alberato, che può essere una cena fra amici. Le idee nascono spontaneamente quando si condivide un tessuto culturale trasversale, e questo è un altro punto incredibilmente importante all'interno di una comunità scientifica. Questo è quello che cerchiamo di fare all'interno del Dipartimento, è quello che proveremo a fare in futuro, e è quello che cerchiamo di fare anche gestendo il progetto Vitality, perché il Dipartimento è il centro di spesa di questo progetto. Vi ringrazio tantissimo per questo invito, ringrazio tantissimo il professor Sauvage e vi auguro una buona serata. Grazie.
Grazie, noi siamo molto contenti ovviamente del supporto dell'Università, ringrazio i molti colleghi che sono presenti qui stasera. Tra questi mi piace ringraziare il direttore generale dell'Università, la dottoressa Anna Vivolo, senza il cui prezioso aiuto e coordinamento di tutta la macchina amministrativa mai sarebbe stato possibile gestire un progetto così grande e così ambizioso. Bene, senza ulteriore indugio vi presento il professor Sauvage. È persona squisita. Se voi poi avrete occasione, anche al termine di questo piccolo incontro, o anche domani, se volete, nei prossimi giorni, lui si trattiene con noi fino a tutto venerdì. Venerdì pomeriggio saremo a Terni per fare un nuovo incontro pubblico, sempre dell'Università, con la presenza che l'Università ha a Terni. Vorrei dare un piccolo consiglio, diciamo, operativo, siccome so che c'è il sole, la luce del sole. Allora, c'è qui un'ampia zona sulla sinistra che è difficile da popolare perché c'è il sole contro. Allora, io suggerirei o vi spostate sull'ala, diciamo così, destra delle sedute, potete occupare anche le sedute qui davanti, che sono un po' più in ombra, oppure un pochino più indietro. Chiaramente, poiché come è noto la Terra ruota, piano piano la situazione migliorerà, oppure la meteorologia può aiutarci se arriva qualche nuvola. Però, come diciamo io insegno all'università, prevedere il futuro non è facilissimo, quindi a vostro rischio e pericolo. Vi presento il professor Sauvage. Il professor Sauvage è francese, ha studiato a Parigi e ha avuto la possibilità, la capacità e anche la fortuna di studiare con un altro premio Nobel, il professor Lehn, da cui ovviamente ha avuto delle ottime indicazioni. Dopodiché si è trasferito all'Università di Strasburgo, da Parigi, e nel 1983, quindi parecchi anni fa, ha compiuto insieme alla sua equipe un esperimento molto importante. Questo esperimento, di cui poi sicuramente ci parlerà, ha a che fare con la manipolazione di oggetti che per noi potrebbero essere familiari. Pensate, che ne so, una ciambella o un cubo, o pensate a una struttura a croce, no? Come se fossero fatti di metallo. Noi sappiamo incastrarli, un gancio lo possiamo legare a un altro gancio. Possiamo creare per esempio una catena. Una catena è fatta di anelli di metallo. Questo lo sappiamo fare. Quello che il professor Sauvage ci ha insegnato a fare è a fare questa cosa con oggetti che sono molto molto piccoli, molto più piccoli di quelli delle dimensioni a cui siamo abituati. Per esempio pensate una catena, come se fosse la catena con cui magari si lega un animale, questa catena però i cui anelli hanno un bordo fatto di un singolo atomo, cioè sono una piccola corona di atomi. Ora gli atomi sono oggetti molto piccoli, delle dimensioni dell'ordine di 10 alla meno 10 metri. Quando ne mettete insieme un po', una decina di questi occupano uno spazio che è dell'ordine di 10 alla meno 9 metri. Questa lunghezza, 10 alla meno 9 metri, si chiama nanometro. Da qui la nanoscienza, cioè la scienza fatta di strutture che hanno queste dimensioni. E quindi le nanomacchine, che è diciamo, il motivo per cui il professor Sauvage è famoso, l'invenzione delle nanomacchine nasce proprio da questa capacità di manipolare, cioè di costruire oggetti che hanno queste dimensioni così piccole. E uno dice, vabbè, ma che servono? Sono una curiosità. Beh, in realtà no, hanno tantissime applicazioni. Sono strutture che non esistono in natura, ma che noi costruiamo. Quale può essere il vantaggio di avere una macchina così piccola? Beh, per esempio, ha il vantaggio di rivoluzionare la medicina. La possibilità di costruire dei farmaci sul posto, oppure di somministrare certi farmaci piuttosto che altri, oppure pensate domani dei piccolissimi robot che possono eseguire delle operazioni su singole cellule. Ecco, questa è una prospettiva estremamente affascinante. Mi piace dirvi questa prospettiva. 1983, la prima evidenza che si possono costruire delle catene di atomi fatti così. 2016, siamo a 20, dunque da 80 a 2006, 30 anni dopo, il premio Nobel, il riconoscimento per questo importante risultato. La scienza va lenta, ma sa riconoscere i propri talenti. Lo dico perché qui ci sono tanti miei colleghi che hanno fatto cose importanti che non hanno ancora avuto il premio Nobel. Quindi però, pazienza, se avete fatto qualcosa di importante, prima o poi verrà riconosciuto. Dal 1983 questo lavoro ovviamente è andato avanti, il gruppo del professor Sauvage è cresciuto, la sua fama è cresciuta, nel 2016 ha ricevuto il premio Nobel insieme a altri due colleghi, di cui poi magari il professor Sauvage ci parlerà, e è nata questa branca della nanoscienza, la nanoscienza è diventata una scienza importante, che è quella della costruzione delle nanomacchine, che è un pezzo del progetto Vitality su cui anche noi stiamo lavorando. Bene, senza oltre indugio, io vorrei chiamare il professor Jean-Pierre Sauvage, vieni qui sul palco. Il professor Sauvage parla l'italiano, molto bene, Per motivi tecnici la lezione, la Lectio Magistralis, sarà condotta in inglese, ma vedrete che le slide sono in italiano. E poi alla fine di questa presentazione ci sarà spazio per fare delle domande e anche quelle le potrete fare tranquillamente in italiano o in inglese, come è più comodo per voi.
O in francese.
O in francese, se volete. Jean-Pierre?
I will speak in English at the beginning. Do you hear me clearly? Great. So first, I would like to thank Professor Luca Gammaitoni for the invitation. I think he told you this is my second time in Peruiga. I was here in 2018 with my wife. I'm still here with my wife. The same wife, by the way. And it's a great pleasure, of course. So Luca, let me compliment you on the project, il progetto fantastico, Vitality. And I must say that, so you are a very visible scientist, and so it's nice that such a very ambitious project has a, let's say, a very respected scientist at its head. So I don't know if you see anything. Maybe we should change something here. Yeah, thank you. So I'm going to talk about nanomachines and nanomotors in biology and in chemistry. So it's going to be a mix. I speak a bit of Italian. This is true, I have to confess. You know. But to talk science, I think it's really much better to speak in English. You know, science is kind of a universal activity of mankind, and English became a compulsory language, so we all have to speak English. So thank you to the people of Progetto Vitality, and thank you to your colleagues too who are helping us. I will start with the very important elements of chemistry. So we will talk about molecules, about elements, and I will start by explaining you, or maybe many of you are aware of that, but everything started in terms of science with the periodic table of elements by Mendeleev. And so here you have this Tavola Periodica degli Elementi, and clearly you can recognize some elements. Yeah, one doesn't see anything. So I think I will work like that. So you have hydrogen, the simplest element. You have lots of other species here. Copper, just at random, molybdenum, arsenic, bismuth. So all the elements. And something which is very important, it is almost sure that the universe is composed of these elements and nothing else. And I think it, in a way, you know, it's really intriguing to know that with this table of elements, you have all the components of the universe. So we will do a bit of chemistry, and I will start with very simple molecules, because I'm sure that not everybody is familiar with molecules. So we have the molecule of water. No, it doesn't work here. Water. Water is everywhere. We say, or we think, I mean, when I say we, scientists think that if you have water somewhere in a planet, it's very likely that life occurred. I'm not sure it's really the case, but of course water is absolutely essential as far as life is concerned. Another important molecule is CO2. So CO2, many people think that it's a nasty molecule because the concentration of CO2 in the atmosphere is too high. This is very true. We have to reduce the percentage of CO2 in the atmosphere. But we have to keep in mind that CO2 is also absolutely essential. Photosynthesis, which uses light, and here we have light, which is rather toxic. Photosynthesis is based on light. And without CO2, we cannot grow plants. We have no life without CO2. So there are two important properties. CO2 is extremely useful, and CO2 is extremely dangerous. The third molecule is dioxygen, O2. So we all know that it is essential for us. When we breathe, of course, we inhale dioxygen, very useful. And the fourth molecule I would like to mention is nitrogen. So we have plenty of nitrogen, or some people say dinitrogen, because you have two atoms of nitrogen connected to one another with three bonds. The bonds are the white segments you see between the two blue spheres. And if you have three segments of this type, it means you have a triple bond. And a triple bond means that N2 is very, very stable. If you want to cleave this triple bond, you have to introduce a lot of energy. So extremely stable molecule. Now, two very simple molecules, or at least one very simple, which is benzene. And benzene is the six-membered ring you see here. And there are several ways of representing benzene. But something which is important, chemists at some stage are lazy, and they do not like to draw C carbon atoms each time there is a carbon atom somewhere. So if you are full of energy, you can draw benzene in this way. And if you are more lazy, you can draw it in the other way, just an hexagon, implying that the summits here, the vertices, let's say, of the hexagon are carbon atoms. Cholesterol is a simple molecule, in biology or in medicine. And so this is a representation of cholesterol. Again, you have to keep in mind that each vertex here is a carbon atom. So this being said, now we will start with modern chemistry and surprising molecules at the beginning. So in 1985, chemists in Brighton, in the UK, and in the US, they collaborated, and they were trying to make molecules which exist in space, so very, very far away from the Earth. And they did something very strange. It failed completely, but they discovered that if you have a very powerful laser, and if you shine light with your very powerful laser on a piece of graphite, you generate very surprising molecules. And this one is C60. You have 60 atoms together. It's exactly like a classical football, a ball, classical football. And C60 is about 1 nanometer wide. So let's say the the object is 1 nanometer large. And 1 nanometer, we have to think of what it means, 1 nanometer. And Professor Gammaitoni already explained that it's 10 to the minus 9 meters. But it doesn't speak to us so much. And I will try to, let's say, to explain, how we can envision the nanometer. So the ratio between 1 nanometer and 1 meter, if 1 meter is here, is about the same as the ratio between a small bar of 30 centimeters, 30 centimeters long, and the distance between the Earth and the Moon. So it means a nanometer is really very, very tiny. Sometimes people say that it's good because it's natural. For shampoo, for a yogurt, for whatever, It's natural. It contains only natural products, natural molecules, and thus it's very healthy. Don't believe it. The most toxic molecules are natural. And I think I have a very nice example here, which is the molecule of tetrodotoxin. And this molecule, it's a beautiful molecule for chemists, this one, with 6-membered rings, nice molecule. It is made by bacteria in the liver of a fish, a Japanese fish, the fugu. And this Japanese fish, the Japanese like to eat it. But if they eat it, and if there is a little bit of tetrodotoxin in the liver of the fish, everybody dies. So this is, I think, the most potent, or one of the most potent poisons known. So the lethal dose, it's indicated here, the lethal dose for men is between 8 and 20 micrograms per kilogram of the person, which means that if you eat 1 or 2 milligrams of it, very likely you're dead. I don't believe there are synthetic poisons which are that potent. Now, we have to talk about motions and molecules, synthetic molecules. By the way, do you know how many molecules have been made by chemists? Millions of molecules. Probably 10 millions of new molecules have been made since 1850 or so, when molecular chemistry started. And so we made many, many, many molecules. And among those synthetic molecules, one can say that very, very little are mobile. I'm very sorry for the people who have sunshine in their eyes. So we say, or we can say safely, that synthetic molecules are motionless. They can vibrate, they can distort a bit, they can compress, but basically they do not move in a controlled fashion. They move in a stochastic way, so at random, and this is really for synthetic molecules. Now let's look at biological molecules. If we immerse ourselves in biology in a living organism, we will find exactly the contrary. The molecules move, and they can move in a very important fashion, but they move in a strictly controlled way. And this is totally different for synthetic molecules. And in biology, there is a family of molecules, motor proteins, and those motor proteins move. They can contract, they can elongate, they can rotate, they can even walk. And I would like just to show you 2 examples, because I believe this is really fascinating to see that in biology, relatively simple molecular systems can move. They can undergo motion, as I said, in a strictly controlled fashion. So I would like to discuss with you two examples of biological molecular machines. ATP synthase, which is responsible for the synthesis of our fuel, ATP, we will talk about it in a minute, and kinesin, which is a walker and has an extremely important biological function. So let's look at ATP synthase first. So you know or you don't know that in living systems there is one fuel and only one fuel, ATP. ATP is a relatively complex molecule, shown here. ATP has a very high energy content, and this is the fuel, exactly like petrol or gas that you put in your car. This is the fuel. And now, if you need fuel, if you need energy, you have to consume the fuel. And in order to consume the fuel, you have to cleave a part of ATP. So you will cleave this part. And so you obtain the degraded form of ATP, which is ADP, adenosine diphosphate. This is not so important. But what is important is that in nature, nature is very ecology-oriented, and nothing is lost, nothing is thrown away. So when ATP is consumed to ADP, ADP being the equivalent of CO2 and water, when you burn a fuel, ADP will be recycled. And this is really a very beautiful system. You keep ADP in your body, and you will recycle it to ATP, so that you will obtain again your fuel, you will fabricate your fuel, and this is a cyclic process. It's not a minor process, because everyone here in the audience is, let's say, recycling ADP to ATP, but it's not 1 gram per day. It's about 1/2 of your weight. 1/2 of your weight, which means that for me, I recycle 40 kilos of ATP every day. A very important process, probably the most important biological process. So how does it work? It has been shown, or it has been suspected for years, that the enzyme which allows you to recycle ADP to ATP is a rotary motor. And this has been suspected for years and years. And there was a beautiful piece of work published by some Japanese groups, one in particular, which demonstrated that this enzyme, which recycles ADP to ATP, is a rotary motor. And you have a nice drawing here, I believe. So you have this part, which is a hollow molecule, and you have an axis which is threaded inside, and this axis will spin, will rotate within the hollow part of the alpha-beta unit. And this, what they did was really fantastic. They attached this axis to a very, very large filament, which they made luminescent, and they could observe experimentally with a microscope that when ATP is consumed, or when ADP is recycled to ATP, it's about the same process, you have a rotary motor. So this is shown here. So this is the luminescent filament. You do not see the enzyme because it's in the black region, but you can see that it rotates. And I think it was some kind of revolutionary publication published many years ago, because the people had their doubts about does it rotate or is it fixed. And nowadays, of course, everybody is convinced that ATP synthase is a rotary motor. It's a very tiny enzyme. It is about maybe 30 nanometers high, 30 nanometers. And it functions for everything which lives on Earth. Everything which lives on Earth is based on ATP synthase and ADP and ATP. The most primitive organisms, very, very primitive, the archaeas, billion years ago, they appeared on Earth, and of course the most advanced organisms like mammals and like human beings. There is a nice video which is very, very close to reality. So this is the enzyme here, you see that. And you have a membrane. You know, in biology, you have lots of membranes, bilayer membranes. And so this enzyme is kind of embedded in the membrane. So the membrane is here, horizontal. And the rotor, the axis which is here, will rotate as you make ATP out of ADP. I don't think we need to enter the details, but here we go. ADP is purple, no, is yellow, I'm sorry. You have inorganic phosphate, but this is not so important. But you see that something is happening here, and finally you make ATP, the purple species, and it can rotate very fast. And so for the moment, you and me, we have billions of ATP molecules of this type which are rotating. Do you feel them? You don't. They are too tiny. Another molecular machine which inspired also chemists is the kinesin. And the kinesin has some property. It can walk on a microtubule. Now let's think of the cell. So we take any cell, and a cell, you have a membrane, so it's a closed space. And within this closed space, many things occur. And those things are first making molecules, small pieces of DNA perhaps, small pieces of proteins, hormones, all that. So you have biosynthetic factories in your cells, and those biosynthetic factories make the molecules which are needed for the cell to survive. But of course, those biofactories are located at some places in the cell, and you have to transport the molecules which have been made at the place where they are needed. And for that, you have it's really a transporting process, like a highway or a railway. You have a small enzyme, which is the kinesin, which can walk on a tubule. So you have very, very long tubes of this type and in the cell, and these very, very long tubes are used by the kinesin. The kinesin is this part here. I'm sorry. The kinesin is this part. It's a walker. So it walks on the microtubule. You have a big bag at the back of the kinesin, and this big bag is full of molecules, and these molecules have to be carried from one dot to another dot, and this is the function of the kinesin, which is to carry them from one place to another. It can be very, very far away. It can be 1 micron, and because the cell is in the order of 1 micron in diameter, and so everything works fine. I should say that it seems to be very slow, but the kinesin is not a walker, it's a runner, And if you think that this is about the size of a human being here, when it carries the big bag of molecule at its back, it runs at about 100 kilometers an hour. So very, very fast runner. It's more a runner than a walker. So a very general statement. Molecular chemists are fascinated by biology, and molecular biology in particular. So they have been inspired by biology, and they are very interested in trying to mimic biology and make artificial systems, of course, which are reminiscent of the living world. And we will now talk about synthesis, and in particular, we will use two families of molecules which seem to be totally inaccessible in the past. A catenane, so Professor Gammaitoni already discussed catenano, chain in Italian or in Latin, and rotaxane, so you have a small ring here, and it has been threaded by an axis, and in order for the system to stay intact, you have to attach two big stoppers, one here and the other here, so that the system will never dissociate. And these molecules have been discussed since 1915, so more than a century ago. Chemists were very interested in this hypothetical system. Hypothetical. And we had to wait for some time before they became reality and before chemists could make such molecules. And I will tell you how we made in our group such molecules. And I think it can be an interesting story because you have luck in it. And I think it shows also that it is very difficult to program research in a rigid way. You should leave freedom to the scientists so that they can express themselves with more imagination and creativity. So I was working with my boss at the time when I was an associate professor or CNRS professor. I was working with Jean-Marie Lehn. He was my boss since my PhD thesis many years ago, and he's now a very, very close friend. And we started in a project which was very ambitious, trying to split the water molecule to H2 and 1/2 O2 using light, because you need a lot of energy if you want to cleave the water molecule. And so we were not the only ones in the world. Many people were very interested in trying to split the water molecule and to make dihydrogen. As you know, dihydrogen is the ideal fuel. When you burn it, you generate water. No CO2, no other polluting molecule. And at the time, the hero, really the hero, was this molecule which is here. So you have a ruthenium center here, and three organic parts around the metal. Ruthenium, if you look at Mendeleev's table, it is below iron, and it has something special. It's a noble metal, so very expensive, very rare even, but everybody was using this molecule. And of course, if you succeed, if you can split the water molecule to H2 and 1/2 O2, it cannot be a ruthenium complex which will do the job because it's too expensive, too rare. And this molecule has very exciting properties. When you shine light on it, you generate what is called a photoexcited state. So the molecule at the beginning is very quiet. Then You send a photon to it, it will absorb it and get excited, very excited. And this excited state has something unique. It can at the same time abstract an electron, which means it's a very strong oxidant, and it can also deliver an electron, which means it's a good reductant. A good oxidant will oxidize water to O2. A good reductant will reduce water to H2. So it has everything you need, O2 and H2. It's too nice, the story looks too beautiful. It never worked the way the people had expected, but still the system is very exciting, very interesting. Except for the ruthenium, which is, as I said, a rare metal. So with a good friend working in the US at Purdue University in Indiana, in the US, and we knew each other from the literature. And David Macmillan had started to use copper instead of ruthenium. And of course, this is very exciting because copper is very, very cheap compared to ruthenium. And so that was really a very exciting project. And he visited Strasbourg, and he spent his sabbatical leave in Strasbourg. And at the time, we had made this compound here, a very simple molecule made by Dr. Dietrich Büchecker, whose name is here. And when Dave Macmillan saw the molecule, because he spent a lot of time in our group, he said, well, it's very exciting. We will collaborate. You make the copper containing species, and we will do all the photophysics and all the photochemistry, or at least photophysics. So we made this molecule. You take two such molecules, you add copper 1+, and you make that. Okay, so that was one thing, and we published a couple of publications on photochemical properties of this molecule and related molecules. But I think the most interesting part, at the beginning, we ignored it. And I will try to show you why we think it was very exciting. Now, let's materialize the endpoints of each component here. So the vertical component here with two dots, blue dots, and let's look at the horizontal component, and the two endpoints are red dots. And now, if we interconnect the two blue points and separately the two red points, what do we obtain? We obtain a catenane. It's very simple. You can do it with your fingers. You do that, and now you make a ring here, you make another ring here. It's a catenane. And so that was the beginning of the field. The people in my group were somewhat reluctant because they were more inorganic, photochemists, inorganic. But one of them decided that it was certainly worthwhile changing research topic, and she embarked in the synthesis of catenanes. So I do it again. So two blue dots, two red dots, and now you interconnect the blue dots and separately the red dots. So we published that. It was, I think, very well received by the community. And then we became more interested in topology, or let's say make a knotted species or a catenated species. And so these are some examples of the molecules we made using the technique based on copper. In particular, the trefoil knot was a real challenge, and this molecule has been discussed for decades and decades, but nobody could make it, and we could obtain very good yields of these compounds. So let's talk about something different. And I wanted to talk about topology, or knotted or catenated species, because these species are very, very common in biology. In nature, you have lots of knotted structures, of catenated structures, and so this is what is indicated here. Catenanes and knots are very common in biology, DNA, proteins, viruses. And perhaps the most impressive structure is that of a virus, HK97 virus. You can stay here. You know, it's a virus which is totally innocent in terms of health. It's a bacteriophage virus which likes to invade bacteria with its DNA. It's a DNA virus. And if you look at the structure, and this is a very precisely defined structure, which is here, this structure is a lateral chainmail. Chainmail in Italian, I'm not sure how you say that, but you know, the chainmail is what the knights during the Middle Ages they used to wear to protect themselves, you know, and fight with their big swords. So it's a very effective protection. So this virus is almost not destructible. So now we will move on to molecular machines. And I say it again, that synthetic molecules are basically static, but in biology, motion is absolutely everywhere. Motion is essential, it's omnipresent, and we will see, we have seen already some examples. So why are catenanes and rhotaxanes so interesting in relation to motions, molecular machines? It's quite simple. If you look at a rhotaxane, like the species which is on the top here, you can easily imagine that if you can move the ring from the right position to the left position, you are making some kind of molecular shuttle, shuttling the ring from one place to another, and at the lowest part here, you can have some rotation, maybe some oscillation. A ring can oscillate within another ring, or you can have some rotation in this species here, which is a rhotaxane. And so I think it was pretty obvious that catenanes and rhotaxanes should lead to molecular machines. Molecules which can be set in motion in a strictly controlled fashion, like the biological system we have seen, ATP synthase or kinesin. So let's look at the very first system we made. That's already some time ago. We will not discuss the chemistry in detail because it's a bit difficult. But we have a copper center here. And the copper center has two states, copper 1+, which is brownish, and copper 2+, which is green or blue-green. And something which is important, at the back here, you have a molecular fragment, which can interact with the copper, depending on whether we have copper 1+ or copper 2+. No need to enter the details, but let's abstract an electron to copper 1+. The system will rearrange completely. We will obtain a new species, and if we inject an electron in the system back, it will change again and you regenerate the starting form of the molecule. So that was the very first molecular machine we had made in our group. And I would like now to mention, do I still have a bit of time, or I don't know. No. I do not see closed eyes, but okay, so maybe 10 minutes or so. Okay, great. Thank you. You're very generous. So a system which was published by Peruiga, a Dutch scientist working in the very north of the Netherlands, in Groningen, who won the Nobel Prize with me. So they made a rotary motor, and this is something very special. You take a molecule with a double bond, a C-C double bond, and this C-C double bond can be distorted using light. You shine light on it, and the double bond will twist, will be totally modified. And if you now allow it to stand in the dark, it will go back to the original form. No need to enter the details, but they published a beautiful video, which is really strictly what happens with the molecule. You have a double bond here, you shine light on it, it moves, you heat, it moves again. And the beauty of the system, what is the beauty of the system? It goes always in the same direction. It's a rotary motor. It's not doing flip-flap or whatever. It rotates always strictly in the same direction. And this is the first rotary motor in chemistry. And that was very important. You know, they did many very exciting things with this species, the first rotary motor. Some other people, and in particular Professor Stoddart and his colleagues, they made una navetta, a shuttle, molecular shuttle, by moving the ring from the green station to the red station, and they went back. And they published a very exciting application. By using the molecular shuttle, they could write at the molecular level by moving the ring. Let's say if the ring is here, they will say this is 0. If the ring is on the red station, they will say this is 1. And now they can write by measuring the electronic conductivity of the system. They knew exactly where the ring was sitting, either on the red station or on the green station, and they could erase. So that was really molecular computing, and everybody was very excited about that. But, and there is a big but, molecules are fragile. And so if you do it maybe 1,000 times, you lose 50% of the activity. And of course, you cannot compete with semiconductors, which are basically non-destructible. So I will show you one of the very last systems. We wanted to make a molecular muscle, a molecule that you can contract or elongate at will. And that was a success. So why did we choose the strategy we used? Very simply because if you look at muscles, you do not contract anything. You have filaments which glide along one another. So this is a muscle here, and you see you have thin filaments, thick filaments, myosin, actin, and these filaments can glide, so you do not force distortion, they glide. You go from the elongated form of the muscle to the contracted form, the contracted form, if you want to make it, you have to use energy, ATP, again. And once you have contracted your muscle, of course, you need to relax, and the molecule will go back spontaneously to the relaxed form. So this is in the striated muscle, known for many, many years or decades. And so we thought it would be exciting to do something a bit similar, to have two filaments which can glide along one another. And this is what we did. It's a very complex system, so no need to, to look at the very structures. But what we do is the following. We have the elongated form. We send a signal, a chemical signal. The system will contract. Another signal, it will elongate, etc. So we have a muscle which is 8 nanometers long when it is elongated, and it is 6 nanometers long when it is contracted. About the same proportion as our muscles, 25% in length of contraction. So I will finish up with the last example. So the people have made a drill on trapano molecolare. And the drill is, I think we will not discuss all the details, but we have some fragment here, which is very close to the chemistry of Ben Feringa and his group. And so this double bond here, using light, starts to rotate. It's a rotary motor. And thus, if you can attach such a fragment here onto the membrane of a cell, if you shine light now, you will have the drill, which will do its job. And what is the job of a drill? To make a hole. And if you make a hole, the cell will die. And this is exactly what happens. So they have even taken pancreatic cancer cells, and you mix it with the drill, you shine light a bit, and the cancer cells die. But, I mean, it's really spectacular. Everybody was very excited. But, and there is a big but, if you want to do studies of this type, of course, you start with in vitro studies, and it's important, of course, that the molecular motor can kill cells in vitro. But there is, as I say here, there is a long way between in vitro studies, which are relatively easy to do, and in vivo studies. Because in vivo, you have a soup. You have so many things, so many molecules in the surrounding of the drill, of the trapano, that you cannot easily go from one in vitro experiment to in vivo experiments. I will finish up by telling you that the field is still very active. We can hope for applications. I will not talk about these applications. I have it in Italian here. So we can store information. This is the work of Stoddart. Let's say molecular computing, but there are difficulties. We can have maybe nanorobots, microrobots using muscles, artificial muscles, and many, many other possibilities. There is only one real application, I think, as I mentioned. And I would like to finish up by telling you that I'm the speaker of today, but the work has been done by a very large group of extremely talented people, and my role is modest. So we have the French CNRS, which is a bit similar to the CNR in Italy. So we worked, as a team with CNRS colleagues. We were, in average, 5 to 6 permanent colleagues and friends, I should say. And we had many PhD students, many postdocs. I would like to thank them above all. I'd like to thank my university, Strasbourg, and the European Commission. They have always been very generous to us. ISIS, which is the institute where I moved after I had to retire from the CNRS. I also spent some time, 3 years, part-time, at Northwestern University near Chicago. My mentor, Jean-Marie Lehn, and I studied inorganic chemistry and organometallic chemistry with Malcolm Green in Oxford, in the UK. I have 2 teachers whom I respect very, very deeply. My family, Carmen, and our son, Julien. I would like to thank Fraser Stoddart and Ben Feringa for their friendship. There was never any competition between us, so it was pure friendship. And finally, thank you for your attention. Thank you.
Grazie Jean-Pierre, è veramente molto bella, non so, io l'ho trovata estremamente interessante. Grazie. Vediamo se ci sono domande del pubblico, io stesso ce ne avrei 53. Qualcuno che vuole fare una domanda? Abbiamo anche il microfono lì. Prego, la domanda può essere fatta in italiano o in inglese come credete meglio.
Salve, grazie per la sua presentazione, professore. Just a small question. Is it possible for you to use this nanomachine not just for movement, for robots, micro-robots, but to build a new type of molecules, to manage the atoms, to discover a new substance. Otherwise, in the normal chemistry, it's not possible. This, I know. I am clear. I think this question is clear.
No, it's not at all.
Thanks.
You know, one of the big projects, which is an ongoing project, is to do chemistry with those molecular machines, to build new molecules using such nano devices by bringing reagents close to one another and making things, you know, molecular systems which have not been made as yet. But thanks to the properties, you know, of these machines, which can really push, you know, components one against the other and force a chemical reaction, we hope that what you suggest, you know, will be possible.
Thanks.
Bene, un'altra domanda. Potete alzare tranquillamente la mano senza problemi, prego, in fondo.
Hi, thanks so much for your wonderful presentation. I was wondering, could you speak a little bit about the origin of the unidirectionality in the case of the molecular motors, such as the one from...
The origin of what? I'm sorry.
The unidirectionality. They only spin the one way. Why don't they just...
Yeah. The system is chiral. I'm not sure if you are a chemist. Chiral, you know, chirality, which means that, of course, one movement will be favored over the other. Because you have chiral components, like a helix, you know. Let's say you have a helix, and it's going to be easier to rotate in one direction than in the other. Let's assume you take, you know, a rod, which is helical, and if you glide something on the helix, it will rotate in one direction. Yeah, this is the secret.
Grazie. Altra domanda? Prego.
Salve. E a proposito del primo degli utilizzi che ha messo nella lista, je peux vous poser la question en français. C'était à propos d'une utilisation à propos de sortir des informations, mais c'est une application transversale ou c'est uniquement pour la chimie?
Sur le, you mean storing information or?
Yes, it's something like artificial intelligence application.
Yeah, sure.
Something, yeah.
No, that would be, making new computers. It would, if it were the case, it would be a real revolution. Because everybody knows that, a data center consume a lot of electricity.
Yeah.
And, our computers are heating. They are heating devices. I can touch it. Yeah, it's a heating device. But if you have molecules, it's going to be very, very different. It's very mild. There is no heating. And I think it's kind of a dream today. But I think it's very possible that one day, we come up with molecules which are extremely strong, extremely robust, and you can store information and treat information using molecular systems.
Another is for cancer, pancreatic cancer. There are advancements because it's very a killer cancer.
Yeah, sure.
Yeah.
There are many groups working on that, using molecular machines, which will be able to travel in fluids, in blood, and get close to malignant cells, to cancer cells, and then kill them by using some toxic chemical. There is a lot of work on that. But again, in vitro studies are very, very difficult. Very, very simple compared to in vivo. Because the living medium is so complex that difficult to control.
Si diceva grandi prospettive per lo sviluppo della medicina. Altre domande? Prego.
One here. Thank you for the talk. I have a question. In biology now we have a lot of technology that don't work exactly on motors, but proteins that have some sort of elasticity and produce very interesting ports. For instance, most of the new technology for DNA sequencing, for extra long sequences, relies on such proteins. The problem is that proteins are not very stable and this technology is not that portable, which is the big problem. Do you think that it's possible to engineer some type of molecules that are not indeed the proteins, but have the same feature in elasticity, in size, and so forth, to offer a more portable and stable technology for the future?
I would say, in principle, yes. But it's probably very, very ambitious. To make synthetic molecules which have properties close to the properties of a protein. Nowadays, I'm not sure. It could take time. It could take time. Proteins have a marvelous advantage. They are available and you can do whatever you like with them. But to make such molecules is very difficult. Yeah.
Grazie. Prego, altre due domande laggiù.
Good evening. I want to ask you about this new technology, which are in the previous future the most hoped and useful utilization on our body.
Utilization of what?
Of this new technology for the benefit of our bodies, which are the most hoped aspiration that this technology can be used.
But molecules can be used, of course, in many fields of application. But for the moment, molecular machines, molecular devices, which imply motion, which are not used. I mean, there are no real applications. I'm very sorry to say, I would be happy if we had some applications, but it's not the case.
Sorry.
I was talking about in the future, which is the hope that the progressive of it.
The progress of
Yeah, the progression of this technology. How can will be used in the future?
Well, I try to give a few examples, but we are molecular chemists, so I'm mostly aware of what's going on in the molecular fields, and not so much about other technologies. But nanotechnologies, it's a very wide field, and you have, inorganic particles, metals. It's a very wide field. I can talk about my field, which is molecular, and unfortunately, there are very limited applications today.
Grazie. C'è un'altra domanda qui davanti.
Yeah, so thank you for the very nice presentation. I was wondering, because you mentioned molecular topology, but I didn't understand if you actually fully exploited it in terms of motion. What I mean is that if you exploit the fact that if you have a certain topology, right, you can constrain the movement because either the object conserves its topology or it breaks it. And I was wondering if you can make these compounds more and more selective, playing with the geometry, let's say. So.
Yeah. So we've been working in two distinct fields. The first field, in terms of chronology, we have been very much interested in molecular topology, making catenanes, more and more complex catenanes, making knots, even complex knots. And it's a field which developed also rapidly. So this is one thing. We didn't care, at the beginning, we didn't care about motion. And at some stage, we realized that using catenanes or rotaxanes, which are very simple topologies, we can make molecular machines. We can have interesting motions, unidirectional, store energy, and do things like that, or the nanomuscle. But in a way, this is disconnected from topology. Yeah.
Altre domande? Intanto che ci pensate ne faccio una io. I have a question. Your systems, they, as you showed us, they need some energy to produce motion. So there is something coming from outside. On the other hand, they naturally live in an environment when there is this fluctuation, ongoing, eternal fluctuation.
Yeah.
Is there any way in which these devices could take advantage of these fluctuations?
Yeah, that's sure. It's very exciting to take advantage of the available energy, which is in the medium. As far as I know, this is done by biological systems, many of them. You have thermal energy fluctuations, and the system moves, and then it is blocked in a certain configuration or certain geometry. And this leads to the motion.
Right.
And we have not done that with synthetic systems. But it has been discussed, by physicists. And there is a physicist who is quite famous, Astumian, in our field, who I think he warned us, we are probably too naive, because we are sending a signal and we believe the molecule will do something, but the molecule is already subjected to motion, solvent molecules, bumping in it and all that. And in biology, this is more or less the way it works. You have the motion and something which blocks the reverse motion.
Yeah. Usually in physics, sometimes they're called ratchets.
Yeah, ratchets.
They say
Exactly. Exactly.
Ok.
Yeah.
Altre domande? Prego.
Okay. Thank you very much for the lecture. So there are no many applications for that yet. But when you started the project, was it pure theoretical interest, or did you have a specific area of interest you would like it to apply into?
To be honest, when we started the project, we wanted to make a catenane, the simplest catenane, because nobody had ever made a catenane in a satisfactory fashion. So that was it. It was kind of, you have a target, the catenane, or a little bit later a knot, a trefoil knot, or thing like that. And it was pure synthetic work, a synthetic challenge, and we wanted to meet the synthetic challenge. And at some stage, we realized that we can also set the molecular systems in motion, and that was also unknown. I mean, nobody had really set molecular systems in motion in a complete way, but it was disconnected. If I answer your question, I'm not sure.
Thank you. And also, so there are biological moving systems. So did you first start making a moving molecule, or was it first realized how the molecules can move and walk in biology? Because, for example, I know that sometimes it's presented that, okay, we have a theoretical systems that works like the biological system, but first chemists made the artificial molecule of this way, and only then they realized how it works in nature. So you already know that it works.
It's almost a philosophical question, and to us very important. I must say that I have always been inspired by biology. And, I used to read Nature, the journal Nature, and I was also reading some biological papers. And so when the work on ATP synthase, the rotary motor, came out, we were very excited. And this coincided with our project on molecular machines, molecular motors. So we were very much inspired by nature.
Okay,
I have to confess. Yeah, very much inspired.
Grazie.
Thank you.
La professoressa Laura Goracci qui davanti. Arriva.
Thank you very much for your talk. Concerning the use of these molecules in vitro assays, can you please comment a bit on the solubility, maybe in water, of these molecules, if there are special tricks that you have to use?
Yeah. For the moment, this is the weak point. These molecules are organic molecules, and I don't believe, they can survive in a biological medium. They will be destroyed rapidly. And so if I had to go on, if I had to propose new projects, I think I would try to work on biocompatible systems. This is the weak point today. There are no biocompatible molecular machines.
Thank you very much.
Very weak point.
Thank you.
I've been impressed by the fact that at the beginning of your research, you were interested in something completely different. And then at some point you changed because maybe you find something more interesting. And often we are told, try until you succeed. But in fact, I've been impressed because you found something more interesting in leaving, letting something away. So I want to ask you, has it been easy, or there has been some resistance or complexity at some point, some pressures, or is it easy to leave something?
Yeah, thank you for the question. It was somewhat difficult because, we scientists are very conservative. And we like to work in the field we know the best. And so the people in my group, when I told them, so maybe we should switch, go from inorganic photochemistry, organometallic catalysis, that was very classical for us, to making new species such as interlocking rings, catenanes, and all that. Many of them were very reluctant. But one of them, my very good friend, Christiane Dietrich Büchecker, she was very enthusiastic from the very beginning, and she was a real genius in terms of synthesis. Anything you could propose to her, with a very difficult synthesis, apparently, she would take it and be successful. And so she did it, and she played a very important role. But if I may add, it also shows that programmed research is stupid. You cannot program research. I think you have to trust the scientists. And if they are bad, they are bad. Okay, it's not because you tell them you must work on this field that they will be more efficient. And I think you should leave, at least to a large extent, freedom to the people who are productive and allow them to switch, from a field to another. I hope you agree.
Grazie. Credo che vada fatta una sottolineatura, questa cosa è importantissima, libertà di ricerca.
Sì.
Facciamo ricerca sulla base di progetti, ma dobbiamo, perché è dove veniamo finanziati, ma dobbiamo avere una grande libertà.
Sì.
Devono avere questa caratteristica di offrire grande libertà di movimento. 99 volte su 100 si parte a cercare una cosa e se ne trova un'altra. È molto importante garantire questa libertà, questa flessibilità nella ricerca, che è nella natura stessa della ricerca. Bene, ci sono altre domande? Prego.
Hi. Sorry, going back to boring stuff. First of all, thanks for the presentation. I was wondering on something regarding the nanomachines.
Yeah.
Most of the examples that you showed, they were performing some type of movement, mostly based on a photo-induced, let's say, interaction, right? So are all nanomachines dependent on light, or there are some other inducers for this type of movements?
So the question is, what is the source of energy, or what?
Yeah, like, for example, the drill or also the catenane at the beginning. All of them were photo-induced processes, right? So are there some other types of interactions that induce these type of movements, or they are always dependent on light?
As far as I know, apart from photochemical, electrochemical, and chemical also, we can use a fuel, a chemical fuel which will be burned, or burnt, in parentheses, not really burned, but a molecule which will be converted to something else, which will act as a fuel, but that's all. There are no other types of, no other means, of setting the system in motion, as far as I know.
Thank you.
And as we said before, I think there is a huge difference between biological machines, which are based on, most of the time on ratchets, as Luca told us, compared to our machines, which are kind of naive. There is still a lot of work to be done before they are really elaborate.
Grazie. Qualche altra domanda? Sì, prego.
Hello, Professor. I wanted to ask you,
Where are you?
Si può alzare in piedi che non vediamo? Grazie.
Okay, grazie.
Since motor, we know motor because by rotation we produce usually energy. Can we use this new type of nanotechnology on living beings like bacteria, fungi, and other species to let them produce by this movement energy from to human purposes?
Yeah, well, it's very ambitious, but it can be envisioned. Yeah, sure, it can be envisioned. But there is still a long way between using these molecules let's say, as a chemist, and using these systems in a biological medium. There is still a long, long way, I'm afraid.
Of course, provided that we keep in mind that energy is conserved.
Yeah.
So we are not going to produce energy from nothing. Bene, altre, prego.
Thank you for the talk. What's about the strength of this system? Because I was thinking about in different environments, this system could rotate more or less.
Yeah.
There is a method to measure the strength of these systems?
Yeah, the solvent is very important. And I know that the rotary motors of Feringa have been studied in various systems. Of course, viscosity is important, but it's important, but it's not vital. Even in a very viscous medium, Feringa's rotary motors still function.
You have tried different environments, so different.
Yeah, not me, but this is the work of Feringa, the rotary motor people.
Okay, thank you.
Bene, c'è una domanda qui davanti. È molto bello, è molto interessante perché ci sono tante domande, di solito non è così, quindi questo è anche il segno di quanto interesse e di quanto sia stato apprezzato questa comunicazione.
Yeah.
Prego.
Thank you so much for your presentation, Professor. Hearing and listening what they are asking to you, everybody here, the comment is that it is interesting that you work in a field and we think, or a lot of us think in another field. For example, for me, I think in a field of biology, application in medicine.
Yeah.
Okay. So the question is not a question, but the thought is, okay, so it is possible that a group of research should have inside not only the professional of one single application, but also the others. Because I am sure that if I see what you discover, I can think how to use what you discover in my field. And when you say that the application you are worried that will need a lot of months or years to be applicable in other fields, is only because there is not a very strength communication between us.
I fully agree on that. I fully agree. I think we should try to work as teams of people, with various fields of competence, and strong interaction. I fully agree. Yeah. But it's not the case as yet. I'm in a chemistry environment, and I have limited opportunity for interacting with biologists or medical doctors. Yeah.
Questo è veramente importantissimo. È anche una delle poche cose che abbiamo tentato di fare con Vitality, facendo dei gruppi molto trasversali, dove abbiamo chimici, fisici, biologi, farmacologi che lavorano insieme.
Sì. È essenziale.
Sì. Molto bene. Bene, abbiamo qualche altra domanda? Se no, se volete, potete anche fermarvi ancora qualche minuto a parlare qui col professor Sauvage. Starà ancora con noi domani e dopodomani. Punto di riferimento è il Dipartimento di Fisica e Geologia, dove lui ha momentaneamente lo studio. Venerdì pomeriggio saremo a Terni per fare un incontro pubblico simile a questo, quindi siete comunque invitati a raggiungerci se volete. Le informazioni le trovate sul sito web di Vitality, su Facebook e Instagram, sempre le pagine di Vitality. Grazie, grazie al professor Sauvage. Vi diamo appuntamento alla prossima iniziativa di Vitality che continuerà ancora per diversi mesi. Grazie.